Hello everyone, I'm Rashmi MG. Welcome to my class. Today let us learn about cell organelles, protein targeting and translocation. Since this is my first class, I would like to give a brief preface or introduction to this topic. So let's begin. This is the content of this slide. It will be dealt step by step, okay? So whenever you remember a cell, you will be thinking of this border and the center one which is having inside the chromatin network. What is it? Nucleus. And it is having a outer boundary, that is nuclear membrane. And also you will be remembering about the endoplasmic reticulum, which are interconnected network. Then you will be remembering about Golgi, which are of 7 stacks. Since there is no space, I am writing like this. And there is a vesicle around the Golgi, right? And then you will be remembering the ribosome, which are of 8 shaped, which are of protein synthesis taking place. And you will be remembering about this mitochondria. You will fancily name it as mitochondria is a powerhouse of the cell. And you will be remembering about what this lysosome, rounded structure, lysosome, which are a suicidal bag. So these are the major or the basic concept you learned in the cell, right? These are known to be what? Cell organelles. To make you know that this ribosome is the protein synthesis site or the protein factory of the cell, right? This ribosome will produce or synthesize proteins. I'll write P as a protein, and this protein are to be transported to different type of organelles here. Why? To perform different functions. Obviously the protein will be having a different type of sequence as its tail. They are having different type of tail. That tail is having sequence. I'll write SEQ, different sequence. That sequence will lead that protein to different type of destination inside the cell. This sequence acts like a pin code for this protein. So what is the function of pin code? It decides the destination of the receiver, right? If you write any message through post, if you send with the pin code, it will be sent to the destination where you want to deliver your message, right? This sequence acts as a pin code. There is a sequence for protein to which the protein should be transferred to the lysosome. There is sequence for Golgi. There is sequence for endoplasmic reticulum. There is sequence for mitochondria also. There is sequence for cytosol also. This sequence of protein will help that protein to transfer to different type of destination inside the cell. That is known to be protein translocation and also protein targeting. Targeting is nothing but that protein is targeted to different types of organelles. Protein translocation means movement of proteins with respect to the sequence. So this is the main gist of the topic. Okay, so let's learn together the different type of organelles and also its function and also the proteins which are translocated to that particular organelle. Okay, I hope you got to know the gist of the topic. So first let us move on with the major protein synthesis factory, that is ribosome. Ribosome is the large ribonucleoprotein. It consists of RNA and protein. We can say it is a protein complex having RNA and protein. It is ubiquitous in cell. I'll let you know why it is ubiquitous. Ubiquitous means it is present all over the cell. It is present inside the cytoplasm. It is present inside the endoplasmic reticulum. Remember, the ribosome which is attached to the endoplasmic reticulum is known as what type of endoplasmic reticulum? Quick revision. It is a rough endoplasmic reticulum. So what is the function of ribosome? It translates genetic information stored in the messenger RNA into the polypeptide. If you remember the central dogma of the molecular biology, there is DNA, RNA and what? Protein. The DNA to RNA process is known to be transcription, and mRNA to the protein is known to be translation. The second step of central dogma of molecular biology is dealt by ribosome. Approximately the structure is globular, as it you will write A shaped. No, it is globular in structure. And also if you look inside the structure, there is a large subunit and a small subunit. The ribosome is made up of two subunits, large subunit and small subunit. The protein which is present in the large subunit is known as L protein, and the protein which is present in the small subunit is known to be S. Here you can see S stands for sedimentation coefficient. It is not this S. You will coming across this S in the next slide. So that is known to be Swedberg. So this point is very important in the competitive exams. So a sedimentation coefficient is what? It is a ratio of a velocity to the centrifugal acceleration. So it is a Swedberg unit. So as I said, the ribosome is ubiquitous in nature. It is present inside this endoplasmic reticulum. This endoplasmic reticulum is known to be what? Rough endoplasmic reticulum. The ribosome which is present in the cytoplasm is known to be free ribosomes. Both produces protein of different type and different function. And then how these ribosomes are associated or how it is organized, let us see. Prokaryotic ribosome is primitive type of ribosome, and eukaryotic ribosome is somewhat complex when it is compared to prokaryotic ribosome. Prokaryotic ribosome is 70S. You can see here S, no, that is Swedberg unit. Here 80S. If they ask a difference between prokaryotic and eukaryotic ribosome, you should write like this. Prokaryotic ribosome is 70S type, eukaryotic ribosome is 80S type. The larger subunit of prokaryotic ribosome is 50S and smaller subunit is 30S. Here larger one is 60S and smaller one is 40S. Further, if the 30S are divided into 16S, and here 40S is of 18S, and it is also having 21 protein. Remember, the first point was RNA. Ribosome is made up of RNA and protein. Here 16S RNA is present and 21 R protein is present. Here this is the association or organization of ribosomes. So genes for all these rRNA are organized as a single transcription unit and transcribed by RNA polymerase. RNA polymerase enzyme is function is to transcribe the RNA. Next one is a eukaryotic ribosome. Eukaryotic ribosomes contain two types, as I already mentioned. Cytosolic ribosome is nothing but the ribosome which are present in the cytosol. Organellar ribosome is mentioning mainly about the endoplasmic reticulum. So organella ribosome, like it is present in mitochondria and chloroplast, there are different ribosomes present inside the cell. Mitochondria has its own protein mechanism, and chloroplast is also having its own protein mechanism, where the ribosomes are present. Cytosolic ribosomes are present in the cytosol, and it is of 80S type. The organella ribosomes are of 70S type. Okay. See, the cytosolic ribosome are of two types: membrane bound ribosomes, free ribosomes. Membrane bound ribosomes is especially mentioning about endoplasmic reticulum membrane. Free ribosome is of what? The cytosolic ribosome. Here you can see the ribosomal structure. Ribosomes first they first organize. The large subunit and the smaller subunit are organized here. Then it forms a chain to form a polyribosome. Then it forms mRNA, long loop of mRNA. Then again the larger subunit and smaller subunit will get divided inside a common pool. After that, whenever it's needed, whenever the protein synthesis is needed, larger subunit and smaller subunit come closer to form the mRNA. See, here the same thing is happening. The polyribosome is what? In cytosol, a single mRNA usually has many ribosomes. See, ribosome 1, 2, 3, 4, 5. These form a loop, or this form a chain, that is known as polyribosome. It forms different type of polypeptide chain or a protein chain. Different protein subunit protein unit will form, ribosome unit will form different protein inside the cell. So this is the organization of the ribosome. Location of ribosome. This table will let you know the difference between the membrane free ribosome and endoplasmic reticulum membrane bound ribosome. Membrane free ribosome is a soluble cytosolic protein. Here it is a secreted protein. Membrane free proteins will synthesize extrinsic membrane proteins localized in the cytoplasmic surface, and it also synthesizes mitochondrial protein, chloroplast protein, peroxisomal protein. Endoplasmic reticulum bound ribosome synthesizes integral plasma membrane protein, lysosomal protein, endoplasmic reticulum protein, Golgi complex protein, extrinsic membrane proteins localized to the extracytoplasmic surface. So different ribosomes are producing different type of proteins across the cell. Here, proteins are synthesized on membrane bound as well as membrane free ribosome. Proteins that are imported into the organelles, inside the mitochondria, chloroplast or peroxisome, are synthesized in membrane free ribosomes, that is in cytosol. Proteins that are imported into ER Golgi system are synthesized in ER membrane bound ribosome. Here you can see protein targeting from free ribosome. Free ribosome is targeted to what? Transported to the nucleus. Soluble enzymes form soluble enzymes. It forms cytoskeleton. It forms protein, mitochondrial protein. It forms the lysosomal proteins or peroxisomal proteins. Protein targeting from ER associated ribosome will form to the Golgi vesicles in endoplasmic reticulum also. It forms the membrane proteins for the cell. Okay. Next, protein targeting and translocation. Cytosolic ribosome synthesizes protein, obviously. Now let us see what are these proteins having. The protein having the amino acid sequence. As I said, the protein will be having a pin code or the sequence that is known as targeting or sorting signals. This sorting signals help the protein to transfer or deliver to the particular specific organelle. The protein which do not have the targeting or the sorting signals, they remain in the cytosol itself. As I mentioned earlier, the protein the protein which is having a sequence with it, that will decide the destination of that protein itself. So you remember what I said, okay? So the protein which do not have any targeting protein will be present in the cytosol itself for the further functioning. Next, sorting signal. What are these sorting signals? How can we define this? This can be signal peptide. Peptide is nothing but made up of protein chain, okay, or it is a signal patch. What does it signals? The signal residue is a discrete stretch of amino acid sequence which is called a signal peptide, also called a signal sequence or uptake targeting sequence. These signal peptides are often present in the end of the polypeptide chain as a tail, okay. The signal peptide is often removed from the protein once sorting is done. So remember, if there is a protein having the tail with it, after it is sent or delivered to a particular organ, the sequence will be breakdown from the protein. So that sequence will get off from the from that protein once the sorting is done. This signal peptide is often removed from the protein once sorting is done. Signal patch can be formed by the juxtaposition of amino acid for regions that are physically separated before the protein folds. Here you can see in this structure, polypeptide synthesized with chloroplast transit sequence. So for the proteins which are targeted for the chloroplast is known to be transit sequence, the green color one. Polypeptide synthesized with chloroplast transit sequence and secondary targeting sequence are translocated or transferred to the chloroplast itself. Polypeptide synthesized with mitochondrial pre-sequence. See, the proteins which are to be targeted to the mitochondria, they are known, they are having the pre-sequence which is targeted to the protein itself. Polypeptide synthesized with the C-terminal PTS signal are only translocated to the peroxisome. We can say that these sorting signals are of different type, are organelle, are particular to the different type of organelles, okay. See, import to nucleus is having this sequence, export from nucleus is having this sequence, import to mitochondria having this sequence. So you need not to remember all this, but think or remember which amino acid is more. So import into nucleus is having lysine rich, export from nucleus is having methionine, leucine and phenylalanine. Like this, the major one you just remember, try to remember. Protein translocation. What is protein translocation? It is a movement of protein across a membrane. Protein translocation occurs in three ways. Let us explore that. Gated transport. What is gated transport? The protein are translocated or transported from cytosol to the nucleus with the nuclear pore complex. This acts as the nuclear pore complex acts as a gate. With that gate, the protein will move from cytosol into the nucleus. Function as selective gates that can actively transport specific macromolecules. Then second way is transmembrane transport, where the protein is transported from the cytosol to the organelle with the help of membrane bound translocators. Then vesicular transport, where the transport of protein occurs from one organelle to another organelle with the help of transport vesicle. You can see here, from cytosol to the endoplasmic reticulum is a blue color one. It is a transmembrane protein where the different types of membrane bound translocators are helping here. Then the red one, cytosol to nucleus, what is the type? It is a gated, where there is a gate present here through which the protein will be transported. Then the green one is a vesicular transport, where the vesicular transport majorly functions to transport the protein from one organelle to another organelle. Protein translocation occurred co-translationally or post-translationally. What is this co-translationally and post-translationally? You know the meaning of translation, where the DNA will be transcribed to form mRNA, and it is translated to the protein P. Okay, this is translation. DNA to RNA is transcription. So mRNA is translated to protein. At this time only the protein is translocated, that is co-translationally. Post-translationally means the mRNA is already translocated to protein. We have ready protein here. That protein is translated later after the translation is complete. Once the translation is complete, after that the protein is translocated, that is known as post-translational translocation. If mRNA is still translating the protein and that protein is continuously translocated, that type of protein is known as co-translational translocation. You got to know the difference between it, right? If the translation process is happening and the protein translocation is happening along with it, that is co-translational translocation. If the mRNA completed the translation process and we are having protein much, that protein after the completion of translation is translocating, that type of thing is known as post-translational protein. Let us know the difference in this from this image. What are the different type of protein translocation? Co-translational protein translocation and post-translational protein translocation. Here you can see co-translation. It is still the mRNA is still translating the protein. By the time the mRNA or the protein is translocated to the particular organelle. Here already mRNA has already translated the protein. After that protein is getting targeted. Here this protein is not present here. Here still mRNA is translating the protein. Okay. So that's it for today. Let us learn about this endoplasmic reticulum in the next class. Thank you.